Okay, so um, let's go ahead and let's talk about um, the migration and extravasation of T cells. Okay, so over here we have our lymph node. Okay, and there's an artery feeding this lymph node, of course, and then there's an efferent lymphatic vessel, and then we go back into the blood vessel. Okay, I've made a quick little key here that describes what each of these things is. The blue ones are going to be your naive T cells. This sort of purplish color is going to be your activated T cells. Green one is your dendritic cell, and then the red one is going to be an endothelium. Okay. All right. So feeding the artery, you can see we have these blue naive T cells and these purple activated T cells. And when we enter the lymph node, we exit out of the high endothelium venule. Okay. Thing is, only these guys, only these naive T cells can exit out here. Okay. These activated T cells can never come back into this high, to the lymph node. And there is a reason for that, okay? We'll get to the reason when we start talking about what kind of chemokines and what kind of receptors these guys have. For now, I want you to understand this. Only these naive T cells will come in, and again, out of all of these guys that are circulating, only these naive T cells will come in. These guys will, interact, okay, with what is that green molecule there? That's the B7 from the dendritic cell. What is that? What is that one over there? That's going to be CD28, okay? So CD28 is interacting with B7, okay? So I want you to know. Okay, now CD28 interacts with B7, and now we have this activated, right? We receive signal 1, we receive signal 2. Again, let's just say this is a CD8 positive, but this could be CD4 as well. If it's a CD8 positive, this is an MHC class 1, and this is a T cell receptor, and that's our antigen, okay? We receive signal 1, we receive signal 2. Now we have an activated T cell. In this case, activated cytotoxic T cell. But again, the whole story can be done with activated CD4 or CD8, okay? So now these activated guys are going to leave through the afferent lymphatic vessel. They go through the blood vessel. Once they go through the blood vessel, they can go into the site of infection. Now let's talk about the mechanism of how these guys are able to get into the HEV, okay? And how these guys, okay, these activated T cells, can get to the peripheral tissue. How can they leave? Okay, let's talk about the extravasation of these T cells. So over here, again, I still have this key here for you guys to follow along, but I won't be using this. I'll be using just my brain to go through this. All right, so again, here in blue, we have this naive T cell that wants to get to the high endothelial venule from the artery, okay, from the artery. And we know that only these naive T cells can get it, okay? These activated T cells, they can't come back into that high endothelial venule that go to the lymph node. They can't go back to a secondary lymphoid tissue. Why not? I'll talk about it. So at the level of the high endothelium venule, we're going to get these chemokines secreted, okay? And these guys are called CCL19 and 21. These guys will get secreted, and then they'll go inside of this blood vessel, okay? And they'll sort of be expressed. They'll be expressed within the high endothelium venule on the the lumen side, and they'll be held up by these proteoglycans. Okay, so let's just say this is CCL19 and this is CCL21. This naive T cell, on its surface, it expresses a receptor for this guy. It expresses CCR7. CCR7 can bind to CCL19 and 21. Okay. We also have some adhesion molecules that we know are pretty standard. Okay. So this pink one on the T cell is going to be LFA1, and then this pink one on the endothelial is going to be ICAM1. Okay. Another thing that I want you to remember is that this naive T cell has the L-selectin on its surface, and then on the surface of the endothelial wall, the L-selectin ligand, aka the adressin, is going to bind. Okay. All right. There we go. And then when that binds, that's going to cause that rolling, okay? And then after that, the mitigation, the rolling, and then diapedesis, okay? So exiting out of that actual vessel and then going into, the site that you want it to go. All right? Okay, that's that. Now let's talk about the second scenario. How do these activated T cells leave the blood to go to the peripheral tissue, the site of infection? Okay? At the peripheral tissue, the site of infection, we talked about how we had MPC and we had CXCL8 being secreted during the innate response to get these sort of mast cells and neutrophils to come out. Okay. In this case, we're going to have the CXCL10. That's the chemokine in this case. CXCL10 is going to be the chemokine that's being secreted from the peripheral tissues, and it'll get expressed on proteoglycan in the luminal side of the endothelium, okay? And it's kind of being held up, okay? This proteoglycan is holding up this CXCL10. What is the receptor on the activated T cell that recognizes this? I'm so glad you asked. It's called CXCR3. CXCR3 recognizes CXCL10. Okay, so that's one bind. Look at what's happening next. Next we have the integrins. Okay, so we talked about this. This is what? This is LFA1 on the surface of the T cell, and then this is ICAM1. Okay. Technically, we could also have VFA1 and VFA1 on the surface here, and then VCAM on the surface of the endothelial. You don't have to know that. Just stick with LFA1 and ICAM. Okay. And if you take a look here, it's quite literally the reverse. The ERP selectin will no longer have an L-selectin anymore. That's only expressed by the naive ones. The ERP selectin is on the surface of the endothelial, and then the ERP selectin ligand is going to be on the activated T cell. Okay? And again, all three of those binding, okay, is going to cause what? That rolling, that extravasation, okay, so that diapedesis motion. Okay? Done. And then, of course, here is your key, okay? So again, like I said, the activated T cell will have CXCL3 on it, okay? And that'll correspond with CXCL10 on the endothelium. It'll also have the LFA1 and then the ICAM1, and it'll also have ERP, selectin ligand, and on the endothelium, there'll be the ERP selectin. Okay? The naive T cell will have the CCR7 receptor, which will correspond with the CCL19 and 21 on the endothelium. Again, the LFA1 and the ICAM1 remains the same. The only thing that changes after the CCR would be the L-selectin would be on the naive T cell, and then on the endothelium we'll have the L-selectin ligand. Okay, that's that. All right.